to give his lecture. Thank you.
Thank you very much. And first of all, I'd like to thank organizers for giving me a chance to talk here in this beautiful campus and for the great support. And I was very much impressed by the name of the workshop, that is from single molecule to cell. So I chose 3 different topics that is relevant to the physical process, how the microscopic molecular processes appear in the mesoscopic or macroscopic system when the underlying microscopics are designed with the biomolecular specific interactions. So the first topic I'd like to introduce is the protein rhythm. I mean that the system only consists of proteins, so 24-hour period. This is very, surprising phenomenon, and lots of theoretical questions are remaining. So we are now, enjoying to think about it. So I like to explain this a bit. And please interrupt me at any time during the talk. So I like to keep the schedule in a flexible way. I mean, just we may talk one topic for over 2 days or something like that. So anyway, as the first part further divided into 3 parts. And first, I like to talk about a general 24-hour rhythm. 24-hour rhythm is sometimes called circadian rhythm. And the new phenomenon here, the main focus of the talk is the in vitro protein oscillator system. And we just think about the future perspective. The last part is not yet published. I just only started to think about it. So, okay. So I'd like to begin about general circadian rhythm. And everybody knows that our human has 24-hour period cycle. And this cycle is relatively independent from the day-night environmental cycle. I mean, we know that through the experience of the jet lag. And actually that the rigid experiments were performed by confined some people into a dark box for several days to keep how their rhythm are changed. But that shows that the human has a definite 24-hour period without external stimuli. And this is similar in many other species on our planet. And essentially that this circadian rhythm has 3 distinct features. One is that, as I said, oscillations are kept in a constant condition, environmental condition without external cue. And next, the period is insensitive to temperature change. This feature is sometimes called temperature compensation. Then I like to go into the details about this feature. This is very, different from the usual biochemical reactions. In usual biochemical reactions, that activation energy is about 10 times or 20 times of the room temperature energy. Then that with the increase of 10 degrees, then the chemical reaction rate in usual biochemical reactions speed up in 1.5 or 2 times faster. So this is why that we need to keep our food in the refrigerator. So because the bacterial activity is slow in the cold temperature. But this circadian period is exception that this does not affect by temperature very much. And the other things, even in the case period is insensitive, phase, oscillatory phase is sensitive to the external cue. So we have these two features, and we need to explain these features in both experimental and theoretical way. You know, the physics theory is apparently needed to understand this phenomena. So the first part is that how we know about the general circadian rhythm. And circadian rhythms are studied in many organisms, including bacteria, like Synechococcus is one kind of the cyanobacteria. And cyanobacteria has chlorophyll. It photosynthesizes the molecule. So their lifespan, life cycle is deeply dependent on the day-night change. So we understand this should be natural that the cyanobacteria has circadian rhythm to survive. And these are the eukaryotes. This is a prokaryote. These are the eukaryotes. This is euglena, the green, single cell organisms we can find in every pond. And by enlarging this part, we have these plants, animals, and fungi. It's mold. And particularly the red typed species are deeply studied in genetics and biochemistry. So through the analysis of these species, the common mechanism to drive the oscillations were found. This is a schematic illustration of the common ubiquitous universal mechanism of circadian rhythm. So these species has typically the clock genes, and clock genes synthesize some clock proteins. And this clock proteins is decreased, degraded by phosphorylation marks or something like that. But this protein, works negatively to its own synthesis. I mean, through the suppression of the positive element, and positive element is an activator binding on the promoter of the clock gene, and this element, this factor enhances the synthesis of this element, but this element inhibits the work of this by binding together. And this pair of the positive and negative elements were found in many species, in cyanobacteria, Kaye and Kaisi, and this is mold, and this is insect and mammals here, period 1, period 2, period 3, and clock proteins, something like that. And actually these people found these clock genes and clock proteins in Drosophila, and received a Nobel Prize in 2017 by establishing this scheme.
So what exactly by positive and negative element, how do they disrupt? Or?
So interaction between this and this is not clear, elusive. Sometimes, it may directly, interact, or sometimes indirect through the third elements or something like that. But this positive element binds to the promoter site of DNA near this clock gene region, and enhances the transcription of this element. And this inhibits action of this. So this is a self-negative feedback loop. You mean that the negative feedback loop should generate oscillation. This is a very simple naive case, and this is a bamboo cylinder, vacant inside, and water is poured from the above constantly, and as water is filled gradually, the center of mass shifts upwards. And if this center of mass exceeds the position of this point, then the bamboo cylinder shows the upside down rotation, and all the stored accumulated water will be expelled. And then this vacant cylinder will go back to this position. So this, is the negative effect for the accumulation of water. I mean, this is just a threshold. When the water amount exceeds the threshold, everything is thrown out, and this is from YouTube. I mean, this is called shishi odoshi in Japanese. This is shishi is animal, and odoshi is surprise or threatening. I mean, that is probably used in ancient time by farmers to, make animals go away from their farms. But from the middle century, that this was used as a tool in the Japanese garden to enjoy the sound of the rhythm. Anyway, and negative feedback helps oscillations. And in the next part, I'll talk about the molecular clock, protein clock system. And then we also found the negative feedback loop and positive feedback loop inside the molecule. But I would like to emphasize that the mechanism is different from this biochemical system inside the molecule. So the work of negative feedback is somehow different, but it is necessary for keeping oscillations. Anyway, and this circadian rhythm was found temperature insensitive. And this is a rather old paper in 1957. And this is the fluorescence from this plankton, single cell organisms was measured to show the 24-hour period. And by changing temperature, the period changes like this. This is the, for increasing temperature, period is elongated. This is opposite to the usual chemical reactions. In usual chemical reactions, by increasing temperature, the reaction rates increases. And then the typical time length decreases. But in this case, time length is increased by increasing temperature. So something opposite, things are happening here. So this was called temperature compensation. Not the temperature constant, but the temperature compensation was used because of this feature. And this is the cause of the red tide. If this explosively grows, this will cause the damage in a fishery. Anyway, in the usual biochemical reactions, we have the Arrhenius law, and delta E here is about 10 kT or 20 kT. Here, T naught is room temperature or something in the body temperature or something like that. And with this temperature dependence, this quantity, the ratio between weights in 10-degree difference is called Q10. And this Q10 is written like this. And Q10, because that by increasing temperature, the rates becomes larger. The Q10 is larger than 1 in U0. That's for with this activation energy, Q10 is 1.4, and with this, Q10 is 2. But in many circadian clocks, Q10 is about between 0.9 and 1.1. So some mechanisms works to suppress this Q10 value in cells. And this is a simple model to explain the Q10 in this organism. This is a mold, red mold, growing on the food. And this is called Goodwin's model, Goodwin equation. And so this is a clock gene, frequency gene. And this frequency gene produces the frequency messenger RNA through the transcription, and then through translation, the frequency protein is synthesized. And this frequency protein is transported into the nucleus, and then this should work negatively, not directly, but effectively negatively, to suppress the transcription of its own frequency messenger RNA. So this is a negative feedback. And we have the several rate constants, K1 for transcription, K2 for translation, and K3 for the nuclear transportation. And K4 and K5, K6 are the rates for the decay, degradation. Then the simple, chemical kinetic equations should look like this. So this is the synthesis of messenger RNA. So if Z increases, this synthesis rate is decreased with power of N. So N represents some cooperativity of binding. And this K4 term is the degradation of messenger RNA. And this Y shows the K2 term is a translation, and K5 is the degradation, and K3 is the transportation, and K6 is degradation. And I do not go into the mathematical solution of these equations, but this would be easy to solve this particularly in a numerical way. Just integrate this in our PC. It is not difficult to do that. We can see the oscillations with these equations. So generally, the time-delayed negative feedback generates oscillations. And in this system, negative feedback lengthens the period, and with some adjusted activation energy, the larger activation energy for K1, K2, K3, negative feedback is strengthened in high temperature. Then that the period is elongated, but in high temperature that everything becomes faster. So those tendencies are compensated with each other. That keep Q10 near to 1 in this equation. I should say that our in vitro system, in vitro protein clock system, I discuss next, the tendency is actually the opposite. But anyway, that as an introduction, that we just summarized our present understanding of how the temperature compensation is realized in user cells. And in more recently, that in mammalian case, that the degradation of this part is very temperature insensitive due to some molecular mechanism. I mean, this is the inside molecule compensation because that the phosphorylation reaction is requires the ATP binding and ADP release, and those balance makes this reaction temperature insensitive. Then that negative feedback weakening reactions is temperature insensitive. Then that this strengthening tendency surmounts overall and realizes the temperature compensation is a recent proposal. But anyway, many things are unknown. We just have this toy model, and there are much distance between the real biochemical understanding and this mathematical understanding, and many things are remaining for our full understanding of the circadian rhythm. But anyway, this is a general oscillations. But I like to go into the main topics of the molecular oscillations. So in 2005, the people in Nagoya, mixed three clock proteins, Chi-A, Chi-B, and Chi-C. They are responsible for the circadian rhythm in cyanobacterium. So by mixing these three proteins in a test tube in vitro, by making these proteins synthesized from bacteria, and they have plenty of a big amount of these proteins artificially, and then they mixed them into a test tube and added some ATP and kept them in about 30 degrees Celsius. Then we found the stable 24-hour oscillation. So this is the electrogel phoresis data showing the different proteins are marked on different positions on gel. And the above line is for the phosphorylated Chi-C, and the line below is the unphosphorylated Chi-C. So the level of the phosphorylation, the degree of phosphorylation of Chi-C oscillates with 24-hour period, approximately, like this. So phosphorylated Chi-C oscillates with 24-hour period, and total concentration is not changed. White mark is unphosphorylated Chi-C. So this is the ratio of the phosphorylation. So there is no transcription or no translation in this system, no DNA or no RNA, only proteins they have. So only the protein-protein interaction and ATP hydrolysis is the source of this generation, oscillations. So this is a big impact. I mean that paradigm shift. So everybody thought that negative feedback in transcription translation loops are the common universal mechanism of circadian rhythm. But in this circadian rhythm, not the transcription translation oscillation, but the protein-protein interactions are responsible for these stable oscillations.
So how do I read this? That as they are phosphorylated versus non-phosphorylated, the concentration is oscillating? Is that the...
Right. Populations, the number of molecules are oscillating.
Phosphorylated versus non-phosphorylated.
And this is the comparison between the in vitro oscillations and in in vivo bacterial oscillations. And the period is plotted for different Kai-C mutants. By substituting some amino acid residue in Kai-C, the period is somehow changed. And that change is the same in vitro and in vivo. So this shows that in vitro oscillation could be responsible to determine the oscillation period in cells. So this protein-protein system could be a pacemaker. It is known that there is transcription translation negative feedback loop in cells in cyanobacteria, but redundantly, it has different oscillation mechanism through the protein-protein interaction, and that protein-protein interaction could be a pacemaker for determining the whole system. It is interesting that many, we have the many redundancy in the biological system, but that could be an example. But anyway, because this is a very simple and well-defined system, people would understand this system in a clear way. But there still remains mysteries. Okay, and this oscillation is temperature compensated, temperature insensitive. This is the oscillations for different temperature, and this is the plot for over the period. And this Q10 value is about 1.08. So we can say that this system is temperature compensated. So the mystery of the temperature compensation is already here in this simplified system.
So if there is something like hibernation, the ATP synthesis is sort of suppressed. So will that control the time period, or will that just suppress the clock itself?
If ATP amount is too small, that oscillations disease stopped. But so we need a large amount of the ATP here.
No, let me be more precise.
Yeah.
So ATP concentration, will that control the amplitude or time period, or none of those?
ATP concentration does not affect amplitude or period if we have enough amount of ATP.
No, so if you keep decreasing it,
yeah.
which of these parameters will be affected?
Phase is most sensitive. So if we changes ATP concentration abruptly, then the phase is shift. Not temperature or period is very much sensitive, but the phase is very sensitive.
So
yes.
Earth has 24-hour period. Venus and Mars is different. So are you predicting these same mechanisms could be working on Venus and Mars and be able to adjust to their diurnal cycles?
Of course, that we bring this sample to Mars, that they will show the same. But so some we don't know how that the bioorganisms evolve in Mars or Venus. So if we found some organisms in Mars, then that will show the different period. So that's we can expect.
Venus has lots of light.
Right. So this is 24 hours.
This might work on Venus, but even though the diurnal period is different.
Right. That's right. That's interesting. But cyanobacteria is a very old species. So and they do not show the exact 24-hour period. So approximate. So the 10% difference is allowed. And this is, but the oscillation phase is sensitive to the temperature change. So if we step up the temperature from 30 degrees Celsius to 45 degrees Celsius at this timing, then that in this case oscillation is advanced, like this. So this, I don't, the color difference is not well represented in this screen, but this is the assumption of the constant temperature, and this outer oscillation is the result of the temperature step up. So phase is advanced here without changing the period much. And this is a plot for how much the phase is advanced when the temperature step up it was performed at different timing. So here the time 16 hour is a peak of the dissociation. So this is the dephosphorylation period, and this is the phosphorylation period, and this is around here is the peak. So if we step up temperature in the phosphorylation period here, then that the phase is delayed. But if we step up the temperature in the dephosphorylation period, then that the phase is advanced. And we have the opposite when the temperature is stepped down. So we need to explain this. Yes.
So in Antarctica, it may be close to zero.
Mm-hmm.
In Antarctica, does this still work at 0 centigrade?
That's interesting. So that we may have a very specific organism around the organism. That could be possible. That's interesting. But cyanobacteria we can find everywhere on Earth. So that this is a test tube experiment. So test tube is put on the water bath, and water bath thermostat was changed sometimes. So we can control the temperature in any way in this in vitro experiment. And this is an experiment to mix two different samples oscillating with different phase. Then how the oscillation after the mixing shows. I mean, for example, when we mix this and this almost opposite oscillation, then that the after mixing, oscillations was entrained to this, dephosphorylation process. So this dephosphorylation process is stronger in entrainment, as we show. We can find from this, and this can be more quantitatively plotted by sampling different samples at A, B, C, D, E, F. And by mixing A with A, B, C, D, E, F, this triangle is position of the peak, timing of the peak. And if A is strong entrainment power, then that these triangles should align along this vertical line. But if the other samples has more stronger entrainment power, then that the triangle should be aligned along this slanted line. So A is weak in entrainment here. B is also weak, but C, D, E is stronger. So this gray hatched time region is a timing having the stronger tendency for the entrainment. This must be explained too. So there are these interesting biochemical experiments showing the logic of this oscillatory system. And this is one thing. And for the molecular biologist, we need to understand from the structure and reactions. And this is a structural biological view, compensating the biochemical view. And this is the constructed from the X-ray and electron microscope image of Chi-C. And Chi-C forms hexamer. And each subunit, each Chi-C monomer has two domains, C1 and C2, in a tandem way. The C1 and C2 are almost homologous to each other. So the very similar C1 and C2 domains are tandemly connected in each subunit, and 6 subunits assembled to form this ring-like form, hexamer. And phosphorylation takes place on C2. Here, that the 431st first serine and 432nd threonine are phosphorylated and dephosphorylated. And this can be found here near the interface between two subunits in C2 ring. So in total, we have 12 sites to be phosphorylated. So ATP binds, this is ATP, and the phosphate group is transferred from ATP to serine or threonine. This is around a few angstrom distance. Okay, this is the mechanism of phosphorylation and this phosphorylation. And the next question is how this timing is coordinated. And this is rather not so accurate, but not high resolution. But this is an electron microscope image of how the Chi2 dimer binds on the C-terminal tail of C2 ring. So Chi-A dimer binds on the C2 tail of the C2 ring, and they pull down the tails to change the structure of C2 ring here. So the biochemical analysis shows that the Chi-A binding enhances phosphorylation, and Chi-A unbinding promotes dephosphorylation. So in a phosphorylation period, Chi-A should bind on Chi-C from the C2 side. And Chi-B can bind on C1. So Chi-B monomer can bind each subunit C1 domain. So 6 Chi-B monomers can bind in dephosphorylation phase. And further, that the Chi-A dimer binds on Chi-B monomer here. So in total, 12 Chi-A molecules can bind on the C1 side. So we have the transitions between them. In phosphorylation phase, Chi-A binds from C2. And dephosphorylation phase, Chi-B and Chi-A binds from the C1 side. So this switching, this transition, should be caused by the structural transition of Chi-C. The structure of Chi-C should depend on the phosphorylation level. So that induces the allosteric structure transitions of Chi-C. That switches the kinetics of Chi-A, Chi-B binding, on which side Chi-A binds or Chi-B binds. Is determined by the phosphorylation level. So we need some, feedback mechanisms inside Chi-C molecule. I mean that Chi-A, Chi-B binding and binding kinetics should depend on the Chi-C structure. And the Chi-C structure should depend on the phosphorylation level. And the phosphorylation level is determined by Chi-A binding. So we have some Yes.
So I'm confused. So the phosphorylation is caused by the binding, ATP binding causing the phosphorylation or dephosphorylation of Chi-C.
Phosphorylation is caused by ATP binding.
To Chi-C.
Yeah, on C2 side.
Yeah. And then as the phosphorylation happens through ATP binding, then it triggers its binding to Chi-A and Chi-B. Is that what you are telling?
No. So to promote, enhance, to process the phosphorylation in C2, Chi-A should bind.
Oh, so the phosphorylation is also promoted by Chi-A binding to.
That's right. Chi-A is necessary for promoting.
Even if ATP is present, the phosphorylation is still dependent on the Chi-A binding.
That's right. If Chi-A concentration is small, there's no phosphorylation process in usual condition. So okay, so the logic is somehow complicated, but we have the structures data and the reactions data and the biochemical logics of oscillations, and we need a unified ground theory. But nobody knows the ground theory, but that's enjoyable topics, I guess. So this is
So I have another question. Where does this periodicity come from? I mean, you showed earlier the concentration of Chi's. So for a given concentration of ATP, and then if the Chi-A binding to Chi-C is very important, then the concentration of phosphorylated or dephosphorylated, where do they generate this periodicity of, say, 20? What?
So the origin of the oscillations must be explained. And then the stability of the oscillations must be explained. I mean that the mechanism should not be too sensitive to the parameters or some details. And the temperature compensation must be explained. So these things is questions. So, okay, and people are still arguing about that. So I do not have a definite answer here today. I will show my own solution, but it is not accepted by everybody. But anyway, so it is now the people are interested in. So we need theoretical model to explain this, because this is a question of the quantitative physics from the molecular perspective. And the further, this is a busy slide, sorry. And this is an evidence for the allosteric structure change of Chi-C. So this is the phosphorylated sites, serine and threonine. Serine 431 and threonine 432 here. And we have helix here. And this is the case. Threonine is replaced by glutamic acid, glutamate. So this has a negative charge. So this is a mimic of phosphorylated threonine, because this phosphorylated threonine is not stable, ready for the crystallization. That this was used as a model for the phosphorylated threonine, because the volume is similar to the phosphorylated threonine and negatively charged. So this glutamate phosphate 2 was used here, but essentially that nothing is changed much. And when threonine is phosphorylated, this forms a stable crystal. This is X-ray structure, and then this helix is broken to become coil, and the bonding direction of these parts are different and bring this part, and so that some long-range molecular deformation could be induced by the phosphorylation of the threonine here. And after selling, phosphorylation selling, after threonine is dephosphorylated, coiled structure is not changed. So we have the two structure states. Just this is our notation. X equal 1 is a state with helix here. And X equal 0 is the structure with coil here. So we have the allosteric transition of chi-C between these two states. And our suggestion is that this is the phosphorylated phase, phosphorylation phase from the dephosphorylated state. And phosphorylation proceeds in this way. So the X equal 1 state favors the binding of chi-A from C2 side. And this X equal 0 state should favor the chi-B binding on C1 state. This is a dephosphorylation process. So that the phosphorylation state of 432 and 431 can control the allosteric transitions, chi-C structures, then that changes the chi-A, chi-B binding affinity on C1 and C2 sides, and that changes the phosphorylation, dephosphorylation rates. So we have the feedback loops here. Sorry for this detailed diagram, but I just want to show that we have the detailed crystalline structure to confirm this view. I mean, as a physicist, that we more favor the simple model.
So histidine has a pK around 6 or so. So if you do the experiments at 5, you don't make the salt bridge any. When you change the salt bridge stability,
that's right.
Change the salt mechanism.
Right, that's right. So we need some ion buffer to control the pH. And we have some range of the pH to show this behavior. Right.
So that could be a test.
Yeah. So right, that's right. So negative charge, the local negative charge should be responsible for the structure change. And another interesting point, I should say that C1 and C2 has very similar structures. And C1 also hydrolyzes ATP, consumes ATP. And it is mysterious why the ATP is consumed in C1. The consumed ATP in C2 is used for supply of phosphate group for phosphorylation, but not in C1. So how the free energy released by the ATP hydrolysis in C1 is used is not known. And this is some structure information that, depending on whether that C1 binding nucleotide is ADP or ATP, that the structure is somehow changed. And this change is correlated to the change of this structure is whether coil or helix. So we have some allosteric communication between C1 and C2. So ATP hydrolysis may part of this process. This is another interesting point. So things are very complicated, but we need both mathematics and structure biology. That's new field. And this is a surprising experiment. So people in Nagoya's group, Kondo's group, they originally found this chi-ABC oscillations, but recently that they substituted a single amino acid residue on this position, Y4O2, to other residues. Then that period is drastically changed. When Y4O2 is changed to cysteine, in this case, the period is very much elongated from 24 hours to several days. And the same things can be found in vitro and in vivo. And as I said, that the C1 and C2 are very similar to each other. So the corresponding residue here is arginine 170 here. So when we superpose C1 and C2 like this, here the C1 is blue cyan and C2 is yellow. R170 and Y42 is very similar to each other, but substituting R170 does not affect the period at all. So because Y402 is just affects the allosteric communication between C1 and C2, that is important to change the period. So this is a new knowledge. And by changing from the alanine and tryptophan, this is a large volume residue. So the period and the volume of the substituted residue is anti-correlated. So the smaller the volume is, the longer the period is. And the difference here is about almost 10 times from the 15 hours to 1 week. So it's a very drastic change. Oscillation, we saw that oscillations are very stable instead of those small substitutions anywhere, and temperature change or some solution condition change. The oscillations are not very much sensitive, but with the single substitution here, the period is drastically changed. So this is a rather striking phenomenon. So this is in vivo data. The similar anti-correlation can be found. So when the residue volume is small at this position, the allosteric communication between C1 and C2 should be weakened with smaller volume of touching the both C1 and C2 boundary. So weakening the allosteric communication elongates the period. So this is a new logic to think about the logical molecular oscillations. So one possible explanation is that we have some negative feedback structure reaction loop inside a single Chi-C molecule. And by weakening that negative or positive feedback, then that period is changed. Okay, I skip this. So we should go into the theoretical models. So many theoretical models have been considered to explain this Chi-ABC oscillations. And usually that the physics people would not go into the detail of the structure or reactions, actually. That they model the structure, for example, P0, for example, that all the 12 sites are unphosphorylated. And one site is phosphorylated, 2 sites is phosphorylated. And finally, all the 12 sites are phosphorylated. And for this, Chi-A is necessary. And we may have the structure transitions. So this is X=1 structure with our notation. This is X=0 structure. So this is with different structures. I just marked with dash. And this is dephosphorylated. Because the Ka cannot bind on the C2 side with this structure. And then Ka-C shows the allosteric transition again. This is the general assumption used by many theoretical models. Of course, there is some weak probability of allosteric transitions like this. Like MWC model of the allosteric transitions, we have the network of transitions. But the most, the strongest transition should take place here and there. It's an assumption. There is no, confirmation from the molecular structural view, but assumption used by many theoreticians. And then, so oscillations in single molecule is evident from the definition of this model or assumptions. Oscillation, single molecule oscillation is already assumed with this model. I mean, of course, this must be explained from the structure and the reaction view. But if we forget about those details, if we use this assumption, then that single molecule oscillations are assured already. But there are still questions to show or to see that test tube oscillation, because the test tube is a macroscopic system. Cyanobacteria is a mesoscopic system, about 1,000 Ka-C molecules or something like that. But in a test tube, we have the much more 10 to the power of 10 or something like molecules. And they show the average of these large number of Ka-C molecules show oscillations. So this is an ensemble oscillation. So individual Ka-C molecules must be synchronized through some communication. So this was the question, try to answer by many theoreticians. So the physicists more focused on the problem of synchronization than the single molecule oscillation system. But we have I should like to emphasize, we have two levels of questions. First is the microscopic molecular explanation to realize this oscillation. And next is the questions on synchronization of the large number of ensemble oscillations. We have two level questions. And it is interesting to see that such a mesoscopic phenomena can be designed microscopically in form of the amino acid residue. So it's an interesting thing. But okay, so the most accepted view for the synchronization mechanism is the mechanism of Ka depletion or Ka sequestration. Ka is hidden away. I mean, because in experiment, if we have too many Ka molecules in a test tube, then the oscillation dies out. So of course, that if Ka molecules are too small number, then that we do not show the oscillation. But if we have too many Ka molecules, the oscillation phase out. So we have some intermediate concentration of Ka to see the oscillations in this system. So a limitation of the amount of the Ka should be responsible for the synchronization is the most widely accepted view. That is, for example, that if Ka preferentially binds on these less phosphorylated Ka-C, then that many Ka is used up around here, and we have a shortage of Ka here to enhance the phosphorylation in the higher phosphorylation level. then that reactions rates are effectively reduced. I mean, the binding reaction is the multiplication of the binding rate constant and multiplied by the concentration of free unbound Ka. So if we do not have a much amount of free unbound Ka because the Ka is already used here, there is no rest free Ka in a test tube solution, then that this rate is decreased. So this is a traffic jam or congestion that makes the population coherent. That aggregates the population here. So that is the mechanism of synchronization proposed by the Holland group, 10 Wolde and other people. So this is a very elegant mathematical model and explains the experiment of the oscillation dying out with the too much amount of Ka. So this Ka sequestration mechanism is widely accepted. But there is no experimental evidence for Ka preferential binding on these states. And actually that I should show the different tendency. I mean, because that entrainment is the strongest in this dephosphorylation phase. Something, special should happening here, not on this side states. And more thing is that because 12 Ka dimers, Ka monomers, 12 Ka molecules can be absorbed in this state. So this can sequestrate. This can hide Ka and depletes the free unbound Ka. Not this state, but this structure should be found around here. So we have some unsettled questions. So, but with this synchronization mechanism and assume some suitable assumptions for the structural reaction allosteric transitions in single Ka-C, we should explain both single Ka-C information and the synchronization phenomenon, or entrainment, or temperature compensation. So this is the goals of theoretical models. The models should be based on the structural observation and the physical principles. And this should explain the stable 24-hour period. And this should explain the large drastic change of the period with single residue substitution. And this should explain the temperature compensation. And this should explain phase shift or phase entrainment, these things. And we need to develop two-level theories, atomic scale theories, and synchronization theory. So that rest thing is my proposal. And probably that I'm not sure that we can talk in 20 minutes or something like that. But I will show tomorrow if we have something that, particularly the last part, I will talk tomorrow. But anyway, my assumption that the structure state is very important. So Ka-C undergoes the allosteric transitions between x equals 0 state and x equals 1 state. And x equals 0 state, Ka does not bind here, C2 side. So it shows proceeds the dephosphorylation. And in X-1 structure, Ka binds here, so phosphorylation proceeds. So D is a measure of the phosphorylation level. D equal 1 when case Chi in the simulated system at time t, the 12 sites are all phosphorylated. Then we have the D equal 1. And if 12 sites are all unphosphorylated, D equals 0. So D oscillates between 0 and 1 in these notations. So, but the individual Chi has different phosphorylation level. And this is the individual Chi structure. So in X equal 1, phosphorylation proceeds, and X equal 0, dephosphorylation proceeds. And this is the probability of the Ka binding. A2, Ka dimer binds on the C2. So this probability increases in X equal 1 structure. And Ka binding probability here, I wrote like this, increases as the structure equals X near to 0. Like this. And that is our assumption, that Ka binding changes the Chi structure, and that can further stabilize the X equal 1 structure. I mean, this complex is stabilized by Ka binding. So X equal 1 structure is positively affected by Ka binding here. But Ka binding is positively proceeded with this structure. So this is the positive feedback. If the structure has X equal 1 state, then Ka tends to bind, and that makes this structure more stable. And the similar things, if Ka binds on this side, then it stabilizes the X equals 0 structure. This is another positive feedback. But we assume that the phosphorylation destabilized the structure. So with X equal 1 structure, Ka binds here, and phosphorylation proceeds, but that destabilized X equal 1 structure. Because with the X-ray structure, that if the S and T sites are phosphorylated, it should show the X equals 0 structure. So the most stable structure is X equals 0 when all the S and T are phosphorylated. So proceeding of the phosphorylation destabilized the X equal 1 structure. So this is a negative feedback. X equal 1 structure emphasizes the Ka binding that promotes phosphorylation, but that destabilizes the original X equal 1 structure. So this is a negative feedback. And the similar things for the dephosphorylation. So X equals 0 structure unbinds Ka from here, but and that unbinding proceeds dephosphorylation, but that destabilizes the X equals 0 structure. And another thing here is that the C1 ATP hydrolysis should become a trigger for the transitions. I mean, we assume that the ATP binding or ADP binding, these ATP hydrolysis reaction cycle is a stochastic kicking of the system. So with ATP binding, X equal 1 structure is stabilized, and X equals 0 structure is destabilized. Actually, that crystal structure with bound ATP is stably found in X-ray crystal analysis. And ADP binding after hydrolysis destabilizes the X equals 1 structure and stabilizes the X equals 0 structure. So ATP binding and hydrolysis and ADP release, that cycle should kick the structure from here to there and from here to this side, or something like that. So we have the multifold positive feedbacks and negative feedbacks and stochastic kicking among them. So this is a baseline of our model to explain the elementary single molecule process from the structure and reactions. So this is an order parameter for the nucleotide binding state. So I wrote that QIKT equal 1 if IC1 domain of the case Chi-C hexamer binds ADP, and this is 0 if ATP is bound. And we assume that the solution has plenty of ATP, so that we neglected the state that binds no nucleotide. So either ADP or ATP are bound on the C1 side, and that changes this parameter, and that gives a kicking effect for this. So that this is the chemical reaction kinetics for the Chi-A binding. X is the concentration of free unbound Chi-A. So HA is the binding constant, and FA is binding rate constant, and FA is the dissociation rate constant. So this is the dissociation term, and this is the binding term. This is just a chemical reaction kinetics, but we define them on each case Chi-C. And in the simulations, we simulated 1,000 or a few thousand Chi-C molecules in the system. Yes.
This equation looks very much like Lauber's original formulation of the kinetics of the Eisenspin. So it's 1 plus m, 1 minus m, and it's flipping back and forth, but it doesn't have oscillation, but you have oscillation, right? What would be the...
This is tangent hyperbolic here. It's just
yeah, so in this case, this was like the magnetization. So 1 plus magnetization, 1 minus magnetization. Okay, and so you have the two terms which are like the probability of finding in upstate.
That's right, that's right.
So in that case, he had relaxation of the magnetization, but you have not relaxation but oscillation. So what could be something different in structure of the equations that are responsible?
Yeah. Important difference is that this is a non-equilibrium system. So ATP is supplied, from the solution.
But in terms of the equation, I'm trying to understand what's the difference here.
This is not much difference. So this is just shows the tendency for the binding and unbinding, and unbinding constant is an increasing function of the X parameter, and this structure parameter. So if structure is near to 1, the binding HA is stronger, and FA is smaller with this, tendency. We can use the different function here, but this is a smooth connection between the x equal 1 and the x equal 0 state. This,
I think you can use just any sigmoidal function. Accidentally,
it looks very similar. So I was wondering, if there is some kind of analogy between the two. But one case doesn't have oscillation, other has oscillation.
Yeah. So of course, it's only this that we have no oscillations, but we have, these multifold reaction loops. Then we have oscillations. And particularly important is the non-equilibrium supply of ATP here. So I will use the analogy of Ising model later more explicitly, but here we just use the sigmoidal in any form. Only a piecewise linear function would be okay. And this is the if Chi-A binding here is regarded as a quasi-equilibrium, we just need to solve the kinetic equations for Chi-B binding and unbinding by changing here. I is the number of Chi-B monomer bound on C1 side, and this I changes with these kinetic equations. And this tendency, plus and minus sign, is opposite here. So the binding is favored for chi b in x equals 0 state. So opposite sign of the sigmoidal here. I mean that the chi b binding, unbinding is extremely slow. I mean the typical rates are 1 molecule per hour or something like that. So it is very slow, yet specific. So it is rather unusual, but probably that the chi b shows some structural transition. And one transition, excited state, can bind on chi c, so that the time needed for this excitation should be 1 hour or something like that. That is probably the reason for the extremely slow chi binding and unbinding reactions here. But chi A binding is rather fast in a time order of the seconds or something like that, like usual biochemical reactions. But chi B binding and unbinding are rather exceptional. That should be relevant to the time scale of 24 hours, but anyway. And we assume the phosphorylation and dephosphorylation, H plus, represents the extent of the chi A binding on C2. So if chi A binds here, phosphorylation is enhanced. And if chi A is unbound, dephosphorylation is enhanced. So this is another kinetic equation for the phosphorylation level. This is nothing special, I think. And rather specific assumption is the treatment of the ATP ACE kicking reactions here. So we treated in our simulations Q as a stochastic variable with some transition from 0 to 1 ATP bound state to ADP bound state. This is the timing of the hydrolysis is caused by a random timing with the frequency, some parameter, frequency parameter, and ADP is released and ATP rebinds with the time duration of delta, and delta again here is we assumed as a stochastic variable. So all the reactions are the usual kinetic reactions, but the reaction constants depend on the structure. That is an assumption, but the reactions are usual kinetic equations, nothing special. But so ATP reactions, we assume that this is a stochastic kicking. So we explicitly simulate it by using the random variables. And then this should be similar to the Ising model analogy. Tangent hyperbolic and inverse temperature is here. So we assumed those binding depends on the structure parameter x, but the structure parameter x should depend on the reaction status. That is an extra assumption, and that closes the loop of feedbacks. This is the long equation, but it shows that if A2 binding probability increases with positive D1, X is increased. So this is shows the effects of the positive feedback. So this is the A2 binding probability acts like an external field on working on the Ising magnet, Ising spins. X behaves like Ising spins, and P here acts like an external field. So also like Ising model, we have tangent hyperbolic functional form. So but this coupling is an assumption on the feedback. So this assumption shows if A2 binds more, then X equal 1 state is more stabilized. And if chi b binds more, then that x equals 0 state is more stabilized with negative minus d2. And this is the, if d is near 1, if phosphorylation proceeds, x equals 1 state is destabilized. This is a negative feedback. And if d is near 0, then that x equals 0 structure is more destabilized. This is a negative feedback, T3. And D4 is the kicking from the x equals 0 to x equals 1 with these Q parameters. So this closes the feedback loops like this. So structure should determine the binding and unbinding rate constants. And binding and unbinding should affect the structure like this, and through the phosphorylation, dephosphorylation. So this works positively or negatively. And ATP hydrolysis can mix them. So with the positive feedback of this D1 term and D2 term, we have stable structure states with x equals 0 and x equals 1 state, 2 states. So this is a switching transition between x equals 0 and x equals 1. And because that this is the negative feedback, destabilized its starting state, and this reaction is slower with the small phosphorylation, dephosphorylation rates. This is a time-delayed negative feedback. So if x equals 1 state is stabilized with chi binding, then phosphorylation proceeds, and gradually that x equals 1 state is destabilized, so that it goes back to the x equals 0 state. So this is the mechanism of our oscillation. And this is the mechanism of our synchronization. I mean that we assume that the total number of the chi A, AT here, is constant. And the chi A exists in a free unbound state, or binds on the C2 side, or binds on the C1 side. And sum of those three states should become constant. So there is a competition of binding to the C2 side or C1 side. So this is makes the communication between different chi-C molecules. This is the only interaction, or some indirect talking through the chi-A binding and unbinding between different chi-C molecules. And this is the similar constraint for the chi-B molecules, but there is no competition here. So competition between these two terms should be the origin of the synchronizations of the many chi-C molecules. If this surmounts the second term, then that many molecules are entrained to the dephosphorylation states. And if this molecules overcomes the last term, then that many molecules are concentrated on the phosphorylation phase. And this is a simulated system, okay, our model. So we simulated 1,000 chi-C molecules in our simulation model, and then the similar amount of the chi-A and chi-B molecules here. And this is an ensemble average oscillation for this black line is the phosphorylation level, D. This was observed by the Kondo's group in 2005. And this red is the average structure, X. And this blue is the Q state. So here is a ADP bound in average. This is ATP bound average. And green is the, differentiation, something like differentiation with this blue curve. This shows the amount of the released ADP. The amount of the released ADP in a short time is large in the phosphorylation phase and small in the dephosphorylation phase. This was also observed experimentally amount of the ADP at different timing was observed. So this explains the experimental data. Anyway, and this is particularly one example single molecule oscillation picked up from this ensemble. So, ATP binds and unbinds occurs stochastically, but structure shows the switching behavior between x equal 1 state and x equal 0 state. And in x equal 1 state, phosphorylation proceeds, so D increases. And the x equals 0 state, phosphorylation decreases. So it's a sawtooth oscillation like this, structure switching transition for x. And we have Q state oscillations. So, I mean that this is the reason why I like to talk about this topic on this workshop, because the model should connect the single molecule to macroscopic large-scale system. And probably that I should continue this in tomorrow to finish our model results and to show the perspective on the more non-equilibrium thermodynamic discussions. So I will stop here today. So by looking at structure and reaction observations and the biochemical logics, we are discussing about theoretical models we need to develop. Thank you.
Thank you, Professor Masaki Sasai, for this wonderful lecture. There are questions from the audience.
You mentioned about the oscillators. So, can this be connected also somehow to Kuramoto's model of synchronization of oscillators?
That's interesting. So we have the synchronization and entrainment, so that the simplest expression should look like Kuramoto model, but I think we have some difference, because that with this Chi-A sequestration mechanism, we have more nearer to some kind of mean field-like picture. I mean, mean field really exists as a free Chi-A concentration. So that entrains the many molecules into some coherent manner. So some different expression from the Kuramoto model would be usable, I guess. So, but the other group shows that the synchronization is caused by the Chi-C monomer exchange between the different Chi-C molecules, not the Chi-A sequestration. So with this model, the Chinese group used the model similar to the Kuramoto model.
So I have another question. That is regarding this number. How many, 1,000? Is this the number of molecules that you mentioned? N equal to 1,000?
N equals 1,000 is near to the in vivo number, but in the test tube experiment, we have more molecules.
Yeah. So if you have very large number, of course, they will probably wash out the oscillations, right?
No, not so. So in a test tube system is a macroscopic system, but we still have a clear oscillations.
Okay. So then the question would be that there is some typical number, as you said, in vivo. So is it possible that it is something that evolutionarily chosen, that kind of number?
Well, because this is a prokaryote bacteria, the cell size is small.
Oh, I see.
Micrometer scale. So, but so this may be the reason why we have the redundancy in the oscillation mechanism, both the transcription-translation oscillations and this protein-protein interaction oscillations. Those two are supporting each other, probably. That's my guess.
I see. Okay, thank you.
Please go ahead.
Hello. I have just one question. So these proteins are temperature insensitive. So my question is that whether these structures are insensitive means with temperature, like heat shock protein or like So these are stable thermally with temperature.
That's an important point. So that people are beginning to measure that. We
will-
have
okay.
some few observations of the by measuring the structure fluctuation, temperature dependence of the structure fluctuation through the X-ray scattering data. But X-ray scattering data is a very indirect measurement by fitting with the small difference with fitting with the mathematical formula or something like that. So nothing is definite. So probably that some people may measure the structure fluctuation, temperature dependence of structure fluctuation through NMR or something more defined spectroscopic way. That would give our answer to our questions.
In
In one of the slides you mentioned that the volume of the sites which get phosphorylated, the period, time period decreases as the volume of the, and then you tried to correlate that with the allostery of that phosphorylated sites with the decrease in the period, time period, correct?
That's the reason why we developed this model. I mean, that this should be represented by changing of these parameters is my claim.
I see.
mean, this is the change in the structural reaction coupling strength. So that should be represented by changes in these parameters is our claim.
So the sites which were chosen, they are across the C2 domain. I mean, so where the sites on which you are correlating your volume. So how, I mean, spread are they in the structures?
So I don't know. So this is only the recent data.
I see.
And only this biochemical measurement was done. And so there are many, residues near boundary, but this position is most sensitive to the period, and we do not know why this is so.
I see.
So more structural observation should be done for answering this. I mean that, this structure picture is paper from 2022. So this is ongoing analysis. Not by, I'm a theoretician, so I just hearing about the results.
any other question from?
Sir, so my question is, what is the possibility that the monomer, not the hexamer, the monomer will show similar oscillatory behavior like the hexamer?
In usual conditions showing oscillations, Cauchy monomer is not observed. Cauchy always formed hexamer. So the isolated Cauchy monomers were not observed in usual oscillatory condition. But there is some evidence that the Cauchy monomer is exchanged between different Cauchy hexamers, I mean by isotope labeling. So this is another question of how this monomer exchange should affect oscillations or something.
Although the monomer is not observed, what do you predict would happen?
Pardon?
You said the monomer has not been observed, but can you predict what will be observed if it is?
That's difficult. So we do not have an atomic model. We only have some intermediate scale model representing the dx or something like that using order parameter for the single molecule. So I cannot answer that question, but that's an interesting question.
Any other question? So if not, let's thanks Professor Masaki Sasai again, and both the speaker, Professor Rieger and Professor Masaki Sasai, for their wonderful presentation. In
So I will talk the rest part tomorrow for this topic, and then move to the another topic.
So come back at 2 o'clock.